Hello ladies and gentlemen, I'm Markus Jansen from LambdaTech, and today I want to give you a presentation about digital assistance for breeders' eyes, AI-based image processing for phenotype analysis. It's about visual inspection and how it goes digital. And breeders frequently look at different things in the breeding process, starting with seeds, overgrowing plants, seedlings, roots, fruits, leaves that have diseases, or complete fields and plots. And then they draw their conclusion from it. And this conclusion drawing can go and can be assisted in a digital way. Whenever you look at something for an inspection, you can also record an image. And these recorded images are processed by software, and information is derived from these images. The derived information in the end supports and improves your conclusion drawing. When you have images and process them, this processing is useful in the inspection process at different levels. The first thing is that you have a documentation. You have images of all your samples, and later on you can also go back to these images and re-inspect them. As a human, you can probably look at a few samples in a short time, but the machine, the imaging and image processing, can do much more. It increases the throughput. When you work with colleagues, the one probably does it a bit different compared to the other. So there is inter-person bias, and this is reduced when you use images and image processing. In the end, the machine only takes the boring part of the job, and it doesn't get tired. When you want to translate images into numerical data, you need a phenotyping software. So there's an image of a plant, and the phenotyping software gets rid of the background and analyzes, for instance, the dimensions of the plants, the colors of the plant, and like here with this indication of the line around the plant, the shape of the plant. And in the end, you get a data table with some parameters, and you can draw your conclusion from the parameters and generate some information out of the parameters. Technically, you first start with image recording. Then the next step is the processing with the phenotyping software, with classical image processing or with machine learning and artificial intelligence. Regardless which method, in the end you extract some information from the image and generate some knowledge. This is an example of some steps that can occur in the image processing. So we have an RGB image. Here is the region of interest where the plant is located. Then the image is converted, for instance, in grayscale in the binary mask. And in the end, you separate the image or the object from the background, and you get a list of the present objects, in this case one plant, but it could be also more than one object. Then the objects are counted, area, shape and color information are derived from the objects. And by the way, that does not only work with RGB images, it can also work, for instance, with fluorescence images, infrared, near-infrared images, hyperspectral images, or whatever image source you might have to get the analysis done. For image processing, we at LambdaTech have two options. The first one is LambdaGrid, the most flexible phenotyping software for user programming. So you can program the image processing by yourself using pre-built functions and a graphical user interface, and this self-programming works without a programming language, so without command line programming. This is a screenshot of the software surface. Here is a library of the functions for image handling, image processing, analysis and data output. Now you see here this enlargement. These are the functions. Here is the graphical surface where you can combine the building blocks out of the library, and each block has a function in the processing. So you start with the image, process it, and in the end you get the parameters. The second option is that we at LambdaTech set up customized analysis workflows with classical image processing or with machine learning and artificial intelligence. Setting up a machine learning process has two parts, a training part and an application part. Starting with image capture for training, you generate some images of your sample material and we provide you a labeling tool and you set labels for the features that are interesting for you. For instance, seeds, roots, shoots and whatever you want to know from the seedlings that germinate here on this paper. Then all goes into a training and the model is predicted. You can inspect the model whether it's already working, probably you have a reiteration of the training, and finally you have a model that you can apply for analyzing your real images coming from the experiment or from your testing series. So you capture these images, the model inference then, and in the end you get information like germination rates or shoot and root size of the seedlings. Let me give some example, and I want to start with the seeds and seedlings, like we had these already in the chart of the machine learning. But I also have some information about plant pathogens and nematodes. I have some example on plant growth and development. Seed germination testing, and I think that's common in breeding processes. You want to check how well the seeds of your cultivars are germinating. You can do that with a paper test. You have moist paper in a tray and the seeds are germinating here, and you check how many seeds have been germinated and are they uniform, are they good or not, and so you do a visual inspection. In this case, we installed a large system with a lot of shelves and an automatic machine that takes the germination trays out of the shelves and puts them in front of cameras, and the cameras record images and analyze them with artificial intelligence as shown before. So we can automatically see whether seedlings have been germinated out of the seeds that are in the tray. So the first image is the seeds before germination, and then germination starts with time and seedlings start growing. Here are the analyzed images. You see these detected seeds and seedlings, and you see the result data. Some cultivars are quick, others are slow, some are germinating more or less completely, others have only only low germination rates. So you see already a quality difference between these cultivars, and of course a quick and complete germinating cultivar is preferable compared to the cultivar that is slow and has only a low germination rate. These germination percentages and the timing of the germination is something that you can also get from visual inspection without technology. So you look at the samples, you take your notes, count the seedlings. But we deliver extra benefit from the LemnaTech data processing. We get developmental data on shoots and roots. For each germinated seedling, the length of the root and the shoot, if present, is measured. And you see it here, again for these 5 cultivars, a comparison, root length growth, and as soon as shoots appear, shoot length growth. This is added data that you would not get when you do a visual inspection, unless you would go there with a ruler and measure each and any emerging root or shoot, which would be very time consuming, of course. So seedling inspection and seed inspection with imaging and image processing is faster than, for instance, visual germination counting. It reaches higher throughput and it's standardizable because you get rid of the inter-operator bias. It can be done manual or automatic. You have seen this large automatic system with the shells, but you can also have a small imaging station on a table where you put in and out the seeds or seedlings on the trays by hand. The AI-based image analysis delivers additional traits that are not accessible to the human inspection. Coming from seeds to leaves and leaves that have been diseased. Here you see a sample from plant pathology, some healthy leaves, some infected leaf that's weed with rust. And we trained the machine learning to recognize rust symptoms, to recognize yellow tissue that comes along with the rust symptoms, and to recognize green tissue. And that can be discriminated and also measured. For each leaf, you get an information about the fraction of green, yellow tissue and area occupied by rust symptoms. That's not only working with wheat and wheat rust, it is also working with other pathogens, for instance with soybean rust. So far we looked at the plants and diseases on plants' leaves, but we can also look at the pest, the causal agent of the disease. In the project we worked with the German Federal Agency of Cultivated Plants, the Julius Kühn Institute, and with an institute from the local university in Aachen, to figure out how to measure nematodes and cysts of the nematodes. Nematodes are living in the soil and they use cysts as propagation stages. Cysts open and the nematodes come out and infect the roots of the plants. So it's important to figure out how many cysts are present in the soil to have an estimate whether this soil can be used for cultivation or not, or whether the plants can be infected in the soil. So cysts are washed out of the soil and then they are counted. So people sit down and count cysts that are here among these soil particles and seeds and whatever can be extracted out of the soil, and that's time consuming. So we set up an imaging and image processing pipeline that does it automatically. So instead of 45 minutes counting, it's only a few minutes for imaging and image processing. And again, you get added value. Each cyst is measured for length and width or area, so you get data that the visual inspector could never get. Inspecting pests and pathogens is also a task that we can address with imaging and image processing. We can also inspect how the plant responds to an infection, because we can look at the plant growth, and that would be something of the next example. We can automate laborious routine tasks by using images and image processing. And the additional traits that come together with, for instance, the counting or the disease rating, give more insight in the process than you would get when you just do a visual inspection. Again, important factor is to avoid inter-operator bias. So looking at growing plants is of course also a thing that breeders do, and here we have an example from the Aberystwyth University in Britain, using a PhenoExpert system where plants are regularly imaged and growth data are derived from the images. So they looked how tall the plant become and how much area of the plant occupy in the images to figure out how the plants are growing. They also looked at yellow area to figure out senescence, and they looked how much water the plants are using during the growth period. All these phenotypic data and the temporal trajectories of the phenotypic traits were used for mathematical fitting and to analyze quantitative traits and detect quantitative trait loci in the genome, because they had this wheat magic population, mapping population analyzed, where they of course knew about the genomic data and could link these genomic data to the phenotypic data to detect the QTLs in the genome. Similarly, the National Institute of Agricultural Science in Korea, also using such a PhenoExpert system, recorded images of rice plants to look for rice seedling vigor. They did phenotypic analysis of inbred lines. You see the pipeline, image, processing, and in the end you have plant dimension data, plant height data, and so on. And again, they could detect QTLs in the genome that are associated with seedling growth. So they carried out a phenome-assisted selection for plant breeding. Plant phenotyping for plant breeding is about linking genotype data to phenotype data. Because to explain how a genome functions, it's important to see how the phenotypes are performing. You can see phenotypic effects of genes, of gene modifications, or of crossings. But you can also see plant environment responses when you expose the plants to different conditions, or you can see treatment or management effects. All these are factors that are important when you are breeding for new plant lines. As a summary and perspective, I want to point out that imaging and image processing enables a comprehensive phenotypic analysis at different stages, at more or less all the stages when you as a breeder do a visual inspection of your plant material. Important points are, for instance, the genetic performance as seen in the last examples, the stress assessments like these examples from plant pathology, and the seed and plant quality like these examples from germinating seedlings. Here you have my contact data to contact me later on, for instance by email or phone. And of course you are very welcome to ask questions to my presentation. So thanks for attention.